Susan, I'm really grateful that you have made the time for this interview in the midst of this busy meeting. And I was wondering if you could begin by briefly telling me about your background and how you became interested in the field of research at first. And you can construe background as narrowly or broadly as you like.
Well, it's fairly accidental, actually. As I grew up, I never wanted to be anything other than a teacher. That's what I wanted to do. And as I went through high school, I decided that math was the area that I would be best suited to teaching, and I was excited about it. I did well in most subjects in school, but I wanted something that I felt was really making a contribution. And when I realized that I was good enough in math that I thought I could pursue a career as a math teacher, it made me very happy, because teaching, making people understand mathematics was something that I thought was very, very important. And I could feel good about devoting my life to it. So that's how I started out. I went to, got a degree and have a Master of Arts in Teaching, and I taught high school math for 3 years until I had my first child. And after that, a friend of ours who was finishing up her PhD in statistics asked me to help her out. She was working with a very eminent biostatistician as a research associate. They needed some help with computer programming and thought that I could do it. She said, we really just need somebody with a math background. We don't need a real programmer. And so I started doing that, and I got interested in the work that they were doing, and I ended up going back and taking a statistics course, and then I took another course, and then my husband said, if you're taking all these courses, you might as well be in a degree program. I'd never really thought of myself as somebody who was going to go on and get a doctoral degree, but in the meantime, of course, I had another child, and so it was a long haul. It took me 8 years to finish my PhD, but I'm very happy that I did, because that gave me the opportunity to be involved with some wonderfully interesting and important research projects. So, like I said, it was kind of accidental.
And how, may I ask, did you end up taking all of that research, all of the statistical background that you developed, and applying it to medical research and to clinical trials, which is seems to be your area of expertise?
Yes. Well, shortly after my husband and I were married, he's a statistician. And he took a position at the National Institutes of Health. So we were living in the Maryland suburbs, and that's where I was teaching. And so when I decided to, and the projects that I was working on with this his name is Jerome Cornfield, wonderful, wonderful statistician. This is actually the 100th anniversary of his birth. He died, unfortunately, too young. So we were working on medical research projects there, and then my husband was at NIH doing medical research projects, and so that's what I wanted to do. It was very, very interesting, and so I just followed along. It never occurred to me to move into any other aspect of statistics.
Right. And Susan, could you describe what your work is today and what the focus of your research is in this moment?
Well, it's pretty varied. Now I'm a professor of biostatistics at the University of Pennsylvania in the Perelman School of Medicine, and I spend half my time as Associate Dean of Clinical Research, which means I am the faculty overseer of the Human Subjects Protection programs in the university. And with the other half of my time, I'm involved with collaborative research projects, several clinical trials. I have one graduate student, and I teach a course in clinical trials. So it's a lot of things to keep me busy.
Wow. So I know that you've done significant research on clinical trial design models, and could you discuss the progression of clinical trial design that you've seen and participated in, and particularly the research you've done to address, you've mentioned the flaws that you saw in the pre-randomization trial design.
Yeah, so it's interesting to remember that clinical trials is a very, very new field. The modern era of clinical trials is generally thought to have started in about the middle of the 20th century. Before then, it was unusual to assign people to treatment by a random process. That was really the beginning of randomized clinical trials in about 1948, 1949. And it rapidly became the standard of understanding how medical treatments work, because you got rid of all of the confounding that you had with observational with observational type studies. And gradually, as the years went by, so I was involved starting in the early 1970s, so it was really at the very dawn of the clinical trial era, and people gradually learned what the problems were, what the pitfalls were in doing clinical trials. At the beginning, for example, if you're doing a clinical trial where mortality or some other serious outcome was what you were looking at, you were trying to see whether you were saving lives by this treatment, it was natural for the investigators to be watching the data all the time to see whether it looked like one was getting better than the other. And then if it did look like that, to be kind of wringing their hands about, well, should we continue with this trial? And very often it became more standard that as soon as the significance level, as soon as the p-value went below 0.05, people would say, oh, we have to stop now. It would be unethical to keep on going. Well, it turned out, this is something that statisticians recognized pretty early, that if you keep looking at your data over and over and over, the chances are that even if there's no difference whatsoever between the treatments, the chances get pretty high that at some point you're going to see a p-value less than 0.05, because clinical trials data are very variable as time goes on, particularly early on when there's not a lot of data in. And so we learned that you have to have a structure for deciding when to stop a trial. And we developed sequential designs and ways to look at our data so that you have to have a more extreme value early on to stop, to be really sure that it's not going to reverse itself, or as sure as you can, as sure as it's reasonable to be. You can never be 100% sure, otherwise we would never do anything or draw any conclusions, because there's always some residual uncertainty. So that's just an example. We learned about the severe biases that can occur if you throw people out of a study because they didn't comply with the treatment, or because of some other reason that you just dropped them and didn't include them in the analysis, and you can severely bias your study that way. So there were a lot of things that we were learning as we went along, and one of the big difficulties in clinical trials always was, how do you get people in them? How do you recruit people for clinical trials? Most of the world doesn't really know much about clinical trials, and if the first time they hear about a clinical trial is when they've just been diagnosed with a serious disease, could be a little upsetting. First of all, you're not really focusing on this separate issue of a clinical trial. You're trying to process the fact that you've just been diagnosed with this disease. And once you do start thinking about it, you may not be so happy about the idea that the treatment, nobody really knows for sure, that the doctor you have picked out says he doesn't really know for sure which treatment you should take, and he or she is planning to flip a coin to decide which treatment to get. And that is something that could be pretty frightening for somebody who's just had a serious illness diagnosed. So in the mid to late 70s, a very eminent professor of biostatistics at Harvard, Marvin Zelen, developed this very clever design that proposed that if you randomize people before you did the informed consent, so that when you did the informed consent, you could tell them, well, we're doing this clinical trial and we're comparing A versus B, and if you agree to be in this trial, you would get B. And his theory was, if you told people what they were going to get, that they would be more apt to agree to participate in the trial than if they had to agree before they knew which they were going to get. And he felt that no matter which one they got, if they knew, it was the not knowing that was the big barrier. Now, I don't know how much evidence there was for that. But so he developed this design, and there's statistically there's certainly nothing wrong with this design, and it could work. The one thing that you would have to do in order to avoid bias is if people did refuse, if they said, well, thank you very much, but I don't want to get that treatment, I want the other treatment, you'd have to include them anyway in the arm to which they were randomized. because otherwise it would be like people selecting their own treatment, and then you'd be back in the observational type study. Wouldn't be a randomized study. So this would only work if very few people refused the treatment that they were given. And so I went to work at the National Cancer Institute in 1982, and there was a lot of excitement about this design. A lot of the investigators, really liked it. They thought this was going to be a way to help get people onto studies that were difficult to accrue to. And I wasn't so sure about this design. And my initial hesitation was because I worried that the design was fine, but in the implementation of this design, an investigator could clearly tailor the presentation of the study, essentially the informed consent process, in favor of the treatment that particular patient had been assigned. So if this patient had been assigned treatment B, the physician trying to, offering the study could say, and if you're in the study, you will get this treatment, and this is really the best treatment for you, and actually say very little about the other treatment. There was nothing to. Now you could have a consent document that had all the right things in it, but mostly what people get out of informed consent, I think, is what, if they get anything out of it, and that's another question, is what they're told verbally in the process. So I was worried that the whole informed consent process could be undermined in this way. I mean, in principle, the person should be told that the treatment was chosen at random. And if there was an informed consent document that was approved by an IRB, presumably the IRB would have made sure that language was in there. But there was nothing to require the presenting physician to say your treatment has been chosen by a flip of a coin. And in fact, I will tell you that when I talked to some physicians about this design, they would tell me they loved this design because they didn't have to talk about the other treatment, and they didn't have to tell the patient that he or she was being randomized. So it was, and they didn't understand. It wasn't as though that they were cheating. They really didn't understand that this was not a design where people could just pick, the treatment that they want, or that they could, that it was a way to avoid essentially a true informed consent. I don't think Professor Zellen believed that, had in mind that physicians would skirt the process and do what I said that they would do. I think he assumed that they would present the study fairly, but there was nothing to ensure that they did. The other thing that happened while I was sort of thinking about this and worrying about this problem was that there were several trials that were ongoing using this design, and it turned out that the rate of people refusing the treatment was not as low as what had been hoped. In fact, and in some trials it was quite high. And it turned out that if as many as 15% of the people refused the drug on each of the two arms, you needed twice as many patients as you would have needed otherwise. Now, what Professor Zellen had said was, even if you do need more people, because you'll be able to recruit so much more easily, you'll make up for that. But it turned out that the trials that had switched to this model had, for the most part, with one important exception, had not doubled, had not increased their accrual to the rate that it was making up for the refusals. One or two trials even switched back to standard randomization because the refusal rate was so high. And again, I think people felt that the problem was that physicians didn't understand, in that case where the refusal rate was very high, they were basically saying you can choose whatever you want. They thought this was a way for people to just choose their treatments, which of course that wasn't the intention at all. So there was some misunderstanding of the design in a lot of different ways. So I decided to write this up, and I wrote a paper that showed what the experience was. And I should say that the trial in which this seemed to be a big success was the very famous trial done by Bernie Fisher and the National Surgical Adjuvant Breast and Bowel Project of mastectomy versus lumpectomy. As you can well imagine, that was a very, very difficult trial to accrue to. If you can just imagine, a woman is diagnosed with breast cancer, and you say, well, we would like to flip a coin to see whether we can just take out the lump or to take your breast off. I mean, that this is, it's hard to imagine a harder trial to accrue to. So they were having difficulties, and they found that when they switched to the pre-randomization approach, the accrual went up not by a factor of 2, but by a factor of 4 or 5. Wow. And they were able to complete this trial. So there was one huge success. But the other trials that were using this were not being so successful, and so I wrote a paper where I documented what the experience had been in the 5 or 6 trials that had used this, plus raised my own concerns about how this could lead to an undermining of the informed consent process. And that paper was published, and it's hard to make connections with with any kind of causal connections. But I do know that the enthusiasm for this design seemed to diminish, whether it had anything to do with my paper or whether it had more to do with it that people were seeing that it wasn't really working. I don't know. But it certainly was probably the first area for me where I saw the interaction between statistical aspects of a study design that I was trained to worry about and the ethical aspects of a study.
Right. And I wonder, could you comment on what your experience was with the background as a statistician to be making an argument about not only the sort of methodological flaws potentially of this design, but also the ethical flaws? In terms of, was that familiar territory for you to be making statements about what you thought was or was not ethically okay for a study?
Well, that's an interesting question. It never occurred to me that that wasn't something for me to think about. All of my experience in clinical trials was that everybody involved in the clinical trial, the clinicians, the statisticians, if there were bioethicists involved, the funders from NIH, and there were always discussions about ethical aspects. What could we certainly the decisions about when to stop trials, that was largely based on, the ethical considerations were essential to that. You didn't want to carry on a trial for such a long time that people could question why you didn't stop it earlier, wasn't the question answered. On the other hand, from a scientific perspective, you needed to carry it on long enough that you could have confidence that the answer you had was the right one. Right. And so there was always this tension. I think anybody who works in clinical trials has to see that, but it is interesting that you mention that, because I remember later on when I was at the FDA, I remember a little tussle between one of the statistical reviewers who worked in my group and a medical reviewer who was very annoyed that the statistician at the review meeting questioned the ethics of a particular study design, and his feeling was, that's not the statistician's job to think, that's my job to think about the ethics. But, I think it's everybody's job to think about the ethics, and in some ways, statisticians are very well positioned to do that because they understand the implications of the inference that's going to be made at the end of the study. And so to me, it's a natural overlap.
And in some sense, if it's not your models that you design for the studies that determine exactly where the equipoise lies, I'm not really sure where you can draw those conclusions from about, it's fair to carry the study on for this long, or, to enroll people in such and such a way. I think that is the job of statistics.
It is the job of the statistician, and even if a statistician isn't necessarily trained in the fine principles of bioethics, they may not have a degree in philosophy, still, there are certain things that are sort of common sense and are reasonable to raise. You may not have the final, I'm not saying the statistician would have the final say, but it's certainly natural to develop opinions about what may be ethical and what may not be ethical.
Susan, do you think you could discuss how you see clinical trial design having evolved since that point, and what do you think the state of the art is? I mean, obviously, trial designs depend on what exactly you're looking at, but do you think that we've made progress in terms of the ability to, produce effective studies that are also, keeping the welfare of patients at the highest priority?
Well, I think you'd probably have some debate about that. I think we have made progress. We've learned how to do better. As I said, we've learned to avoid some of the earlier pitfalls that I think people entered into out of the best of intentions. Wanting to get the right, their goal to get the right answer often led them to do things that would increase the risk of their getting a wrong answer. And so we've learned about those kinds of things. We've learned about not throwing people out of an analysis. We've learned about trying to follow up people, and that it's a big problem if you have a lot of missing data. We've developed methods to try and analyze studies even when you do have some missing data, to try and see how those missing data might have affected the results and how far off you might be. We've learned, as I said before, about study designs for monitoring and how we oversee studies, what data monitoring committee will look at. We have a whole new era now of adaptive designs. Now, clinical trials have always by necessity been adaptive in that, you have to watch what's happening. If you see some unusual safety issue emerging, you have to be prepared to do something about it. You might have to reduce doses. You might have to drop one of the treatments that you're studying. You might have to stop the study altogether. So they're always been adaptive in that sense. And then the sequential designs that said we're going to look at our data, every 6 months, and if we get to this point, if we cross this boundary, then we'll stop a trial. That's an adaptive design. Early on, probably back in the 80s, people started developing other types of adaptive designs in which you would change the ratio of those assigned to one treatment versus the other treatment according to how the data were emerging. So that
pick the winner.
Yes, and actually I should say the play the winner rule was also developed by Marvin Zelen. I mean, Marvin Zelen has really been an extremely creative statistician when it comes to clinical trial design. So he developed the play the winner rule, and that got many modifications developed in that. But one of them was basically a design called a response adaptive design, where as the data sort of emerge, you start saying, all right, well, now instead of 1-to-1, it's going to be 1.5-to-1 or 2-to-1 or 3-to-1. Now those designs, while they have a certain appeal, right, because you can show that at the end of those studies, which on average are going to be a little bit bigger probably than other studies, but you're going to have many more people be getting the superior treatment by the end of the study. I think there are many, though, who feel a little bit uncomfortable about the idea that you've designed in the study to figure out which is looking better, but you're still assigning some treatments, some of the treatments that look worse to some of the people. So, if you're almost sure that treatment A is better, then that you have still assigning 1 out of 4 people to the worst. There was something that made a lot of people feel uncomfortable. And the people who promote those designs will say, what's the difference between that and if you keep it 1-to-1 all the way along, and the last person into that study, and maybe it's it's pretty clear by then. And this is a hard thing to adjudicate. I think most of us who are used to the standard designs think of the progress of a clinical trial as one where we say we don't know anything at the beginning, and we say we do not know anything until the end when the results are released. So basically our knowledge is 0, 0, 0, 0, 0 all along. So we don't so much have a problem with who's being randomized to what toward the end, because our knowledge is still zero. We do have this data monitoring committee up here watching to make sure things don't get totally out of hand, but until we know, we don't know. So it's zero and then it goes up to one at the time. Whereas the other one, you are saying that you know something by the design as it goes along. You know enough that you're doing 2 to 1. You know enough that you're doing 3 to 1. So practically, does it make a difference? I don't know, but that's one reason I think that these designs have not become more popular. But the new version of adaptive designs are different. They're intended to, in the earlier phase of studies, they're intended to allow for a lot of variability in which doses you're studying, or even which combinations of treatments you're studying, or even which populations you're studying, and to be able to shift things so that the theory is that you will be able to make a decision about which treatments to move forward with faster, and you will get rid of treatments that look like they're not going to be effective sooner. We've had a speaker at this meeting yesterday, Don Barry, who's been in the forefront of this kind of a design. And I don't think there's too much controversy about those designs, although I'm not sure everybody's convinced that you really do get the right answer faster. But there's a lot of people using these designs now. There are other kinds of adaptive designs that are intended to allow a study to enlarge if you get partway through and it looks as though this may work but not quite as well as you thought in the beginning, and the study you designed wasn't big enough, and so you make the study bigger. And so there are a lot of different types of adaptive designs. That's sort of what's new in clinical trials. People looking at these designs to see whether they're going to help us get to the right answer faster.
To me, the design of clinical trials from a modeling perspective looks like an ethical quagmire. I mean, so where are you turning to get guidance to see what model looks most persuasive to you, or what you want to work on yourself?
Well, the kinds of designs that I have been talking about, I mean, there are certainly ethical issues every time you design a clinical trial. So when you're talking about when should a trial stop, there's a lot of controversy about how much evidence you should need before you stop a trial early, before its intended closure. And there are those who feel that you should almost never stop a trial early. Once you start, you should just keep going unless you have an extraordinarily strong effect. Others feel that the clinical trial is a designed experiment, and if you get to a point where the probability is very, very low that you're going to do anything other than conclude that this treatment works, then why is it ethical to continue to randomize people to possibly the inferior treatment? And so there have been debates about trials that have, say, ended with a p-value of something like 0.000001, and it certainly convinced everybody about the treatment effect, but many people feel, well, that trial should have stopped sooner. And the other argument is, if we stopped it sooner and we didn't get all those zeros before the 1 in the p-value, we might not have really had persuasive enough evidence to change people's medical practice. And that's a hard thing to prove one way or the other. But there are very different perspectives on how much data you need. And there's no right answer to that. There's no right answer to that.
I guess I imagine one source of information or one source of influence on those decisions is the people undergoing the trials themselves. And I know that you've done extensive work with the early design of AIDS trials. Could you talk at all about how the desire of those people to be in certain kinds of trials affected your thinking about the design of trials?
Yeah, the AIDS situation was very, very interesting and a very gratifying part of my career. I moved from, I was working in cancer trials from 1979 through 1988, and in 1988 I went to the National Institute of Allergy and Infectious Diseases as the first chief of the biostatistics branch in the Division of AIDS. So it was a new division that had been formed to develop large-scale clinical trials for AIDS treatments. AIDS was people were newly aware of AIDS in the mid to late 1980s, and when I joined NIAID in 1988, the only approved treatment for AIDS was AZT. There were new trials of a couple of new antiviral drugs had been started, and there were trials of treatments and preventive mechanisms for the opportunistic infections that ultimately were what killed AIDS patients. And it was a time, but by the time that I came in '88, the community groups, the patient activist movements, were in full force. They were very angry that there had not been enough attention paid to AIDS, that not enough resources had gone into dealing with the epidemic, and they were starting to be heard, and they were feared. They were feared, and the investigators in the new AIDS clinical trials group that had been formed were very fearful that the demands of the activists were going to prevent them from doing the kind of clinical trials that were ultimately going to give the answers that they needed. They felt that these people were misguided. So, but it was very political. Nobody likes to see a headline in the newspaper saying that this group, whatever, says that these people are murderers, and that's the kind of thing that was happening. So. About a year after I joined, I went to an international AIDS conference, and the AIDS activists, ACT UP in particular, were there, and they were quite vocal, and they were handing out documents that they had put together that described how they thought the clinical trials programs of the NIH needed to be changed to be more humane and to get better answers. And so I took one of these documents, and I started reading it, and I was actually quite surprised that most of what was in there sounded quite reasonable to me. In fact, I was surprised. They said, somebody who is in a clinical trial should be able to take other medications that they need for their health. Well, who could argue with that? These are young people who had a life-threatening disease, and if there were other medications that they needed, why should they be told in a clinical trial that they couldn't take them? So there were a lot of things like that seemed perfectly sensible, and that I thought there was no reason in the world that we couldn't accommodate those kinds of things into clinical trials. In fact, even if these were unproven medications, if lots of people were going to be taking these medications together with the antiviral drugs that we were testing, wasn't it important to know what the effect was in that context? That was the real world answer that we were looking for. So there were things in there that I did not agree with. For example, they argue that there should never be any placebo-controlled trials. And I thought to myself, well, we're not going to do a placebo-controlled trial of a new antiviral when we have AZT, which clearly saves lives. Now, we didn't understand at that time how short-lived the effect of AZT was, but certainly in the short term, it did save lives. But there were many other kinds of trials in which if we did not have a placebo, we would never know whether the treatment was working or not. So I brought this back, and we had a sort of a small meeting of statisticians who were working on AIDS trials, people in my group at NIH, people at the FDA, people from the statistical group at Harvard who were the statisticians for the AIDS clinical trials group. And I made copies of this document, and we discussed it, and there was a lot of excitement. And in fact, we actually had 2 members of ACT UP come to that meeting because they had gotten word that we were going to be discussing this document. They said, well, we need to be there. We need to be at that table. So it was a terrific meeting, and there was lots of agreement. And the people who were representing ACT UP were very involved and engaged in the conversation, but they listened as much as they talked. And we agreed that we really needed to have another one of these meetings right away because we didn't get anywhere near through the documents. So we organized another meeting. And at the next meeting, there were more people from ACT UP there, and there were more people from the NIH, people who weren't specifically involved in AIDS trials but had heard about this and thought it was interesting and wanted to come. Some of the investigators started to come. They were pretty brave because at that time the AIDS activists weren't permitted to attend any of the planning meetings of the AIDS clinical trials group. Because the investigators, again, just felt that they were the enemy, that they were going to, all they wanted was all the drugs right away without any clinical trials. And it was clear to us, the statisticians, that's not what they wanted at all. What they wanted was that the best trials to be done. They wanted answers as fast as they could get them, but not at the expense of there not being the right answers. So we had meeting after meeting, and at each meeting there were more people. The first meeting there was maybe 20 people. Pretty soon there were 100 people or more coming to each of these meetings, and they were very exciting. We just exchanged a lot of information, and I think these meetings helped the activists certainly. First of all, they introduced them to a lot of other people, so they had many connections now that they could go to talk to about the design of clinical trials, and they rapidly developed a terrific expertise. When they had come in initially to the meeting, they had a lot of expertise already about the drugs that were, they were learning pharmacology, they were learning mechanisms of action, they were learning about all of these aspects, but now they were also learning about study design issues. And within about a year, the ACTG leadership relented, and they became actually a formal part of the mechanism with different community members sitting on various disease committees and having a formal role in planning the trials. And it was a terrible time, of course, but it was a very gratifying thing to be part of, to feel like, we can work together. This is not an us versus them, and we did work together.
And do you feel like the trials were improved as a result of that collaboration?
Yes, I do. I do feel that the trials were improved. I think the ACTG leadership, the clinicians doing trials, and it was a common. Many of them had grown up, in a system where there were just certain rules that you did with clinical trials. That if things were going to go to the FDA insisted that things be a certain way. Well, the FDA was willing to be quite flexible. So I think we the more we discussed, the more common ground we saw, and people saw that they could move ahead and design trials that would be acceptable. I'll give you an example of something that I thought was particularly interesting. I was involved with a different institute. The National Institute of Mental Health was developing a trial it was intended to prevent the neurological complications of AIDS, and there was a drug that had been developed. I think it was been developed there by NIMH scientists, and they wanted to do a trial where everybody would be on AZT, and they would randomize between this new drug and placebo. So nobody was being untreated, but only half the people would be getting this new drug. So I knew that this drug was sort of a fringe drug, in that the people who were really interested in it, it was a specific group of activists who thought this was really going to be a useful drug, and they were very excited about it. And for the most part, those people did not want anything to do with AZT. They were really looking for alternatives to AZT. AZT was not an easy drug to take. It was toxic, and there were some people treating AIDS patients who were very negative about AZT, who did not want to treat their patients with AZT. So I advised the people at NIMH. I said, if you insist that everybody in the study be on AZT, and I understand that it would be unethical to not let them be on AZT, but there's going to be a lot of people who want to take part in this trial, who want to possibly have access to this drug, who are not going to want to take AZT. And you are going to have a big problem if you insist. Oh no, they said, it would be unethical not to have everybody on AZT because we know AZT is life-threatening. So I said, okay. So they had a meeting with the group of people who were anxious who had been pushing to have this study done. And as I predicted, when they announced what the study design was going to be, everybody would be on AZT, and then pandemonium broke out. People were very angry. They did not want that study design. What I had offered them before, I said we could do a stratified design where everybody was offered AZT, but people who did not want to take AZT could simply be randomized between this drug and placebo, and that people who took AZT would be randomized to the same thing, but they would have AZT as a background. So I said, you encourage everybody to take AZT, but you don't force it. That they thought was unethical. When they heard back from the community at that meeting, One of the leaders at NIMH then said, well, maybe we could do a stratified design. And that, of course, was readily accepted. So that's an example of the kind of compromise that you had to make in a traditional clinical trials environment. You would never think about not having everybody on a treatment that's known to be life-saving, but in this circumstance, you had to, think about who you're and was it unethical not to insist that everybody take AZT? There were lots of people who were refusing to take AZT.
Right, outside of trial.
Outside of trial. So to me, that was an ethical design.
And do you think introducing that stratification, did that confound the variables in any way? Did that compromise the...
No,
No,
because you when you do a stratified design, what you do is you compare the two treatments within each stratum, and then you aggregate. The way it could confound things is if you had what they call an interaction. So that if this new drug only worked in people who weren't taking AZT, but it didn't work if they were taking AZT, or vice versa, or it was even harmful in one group, then you wouldn't be able to aggregate. You wouldn't be able to add up. You'd have to look at it separately, and then you'd probably be underpowered. So people do worry about that in some of these kinds of trials, but it's not usually a problem.
Right. And it's just interesting. It shows the complexity of what it means to be respecting research participant autonomy. That the first complaint that the activists had was that that thought placebos should never be part of trials, and then later on saying, I don't want to be forced to take a medication. I'd rather be potentially randomized to placebo entirely, essentially.
Well, they wouldn't have been randomized to placebo, but they wanted the option not to take the standard drug. Yeah.
That's true.
Yeah. Well, I think that's part of recognizing that not every treatment works. We were very fond of the statement that a medicine cabinet full of 100 drugs is useless if you don't know which drugs work. And I think that is something that the activists, realized very, very quickly.
Right. Speaking of placebos, you've also done a significant amount of work, it seems, to show that placebos, in fact, are necessary and valuable in certain kinds of clinical trial cases. Could you talk about where you've seen that to be true, or what your research in that area was?
Yeah, so most of my work after I worked in AIDS for 5 years, I went to the Food and Drug Administration, and most of my work before I went to the FDA had been in areas of serious disease, cancer and AIDS, where once you have a known treatment that's known to be life-saving or prevent cancer relapse, you don't do placebo-controlled trials or trials with a no treatment control. But at the FDA, I learned that there were many other kinds of trials. We have lots of drugs that are not life-saving drugs but are very valuable drugs. If you have allergic rhinitis and you spend all day long, blowing your nose, and then suddenly there's a drug that can control that. That's a big quality of life factor, and that is a very important drug for you to have. But there's nothing, but your life is not at stake if you are in a placebo-controlled trial of a new drug for allergic rhinitis. A problem in doing clinical trials of many of those kinds of drugs that relieve symptoms but don't have an impact on fundamental health, is that the results are very, very variable for reasons that people haven't figured out. Drugs that everybody believes work, if you do 5 or 6 trials, you will find it seems not to work in some of those trials. And nobody has really figured out why. It's true for drugs, and any symptom relieving drug, pain drugs, allergic rhinitis drugs, antidepressants. It's a sort of a well-known phenomenon. So a problem there is if you want to compare a new drug for allergic rhinitis against an existing drug for allergic rhinitis, and they come out to be the same, you can't necessarily conclude that the new drug is working because that drug, the drug that you think works, might not have been working in that trial. Why? Maybe for some reason the people in that trial, there weren't a lot of allergens around during the time that they were in the trial, or it's hard to know. But because it's very common for trials of this sort to be negative, even when there's a known effective treatment involved, the FDA has learned not to trust active control trials. So they insist for these kinds of trials on at least a placebo. Their optimal design is a placebo and an active control and the new drug. That way, if the new drug doesn't do better than placebo, but the active drug doesn't do better than placebo either, you know that you've got a failed trial. If the drug that you know works is better than placebo, but the new drug isn't better than placebo, then you start to get a clue that maybe this new drug is not effective. But you need the placebo as an anchor, otherwise you don't know where you are. So the question is it unethical to do a trial? You have an existing treatment, and is it unethical to do a trial with a placebo control? And I think when people started to raise this, it was a surprise to people at the FDA because it wouldn't have occurred to them to think there's anything unethical about telling somebody who has a symptom that there's a clinical trial, they might get a placebo, the new drug might or might not work, new drug might have some side effects. We just want to see if it works. You'll be on this these are usually short-term trials, maybe 2 to 6 weeks. You can go off the trial anytime you want. So, for example, if somebody, is really having bad symptoms and they just can't stand it anymore, they just go off the trial. Didn't seem unethical. Nobody's fundamental health is being risked. But there were also a big in the news at this time. This was the late 1990s. Were AIDS trials in developing countries that were utilizing placebos because the regimens that they were testing were very simplified regimens compared to what had been shown to be effective in the U.S. And the NIH and the CDC, who were sponsoring these trials, were concerned that the simplified regimens might not work at all. The more complex regimens that were used in the United States were not implementable in these developing countries. And in particular, these are trials of mother-to-child transmission of HIV that involved starting the mother on antiviral drugs, on AZT, partway through her pregnancy. In the U.S., people come in for prenatal visits. In many areas in the developing countries, that was not feasible. You would not get to the woman until it was time to give birth, and so nobody was sure that it would work at all. And AZT was not a trivial drug. Nobody knew for sure what it was going to do to developing children. Even after it had been successful in the U.S. in preventing transmission, we didn't know whether there weren't going to be side effects down the road in the children. So those trials were originally designed as placebo-controlled trials to see whether, in fact, these were going to have any effect. But there was a huge uproar about that and whether those trials were ethical. So that fed into this whole debate about placebo-controlled trials. And I think people thinking about babies infected with AIDS in developing countries, they didn't want to think about people on 2-week trials for allergic rhinitis. They just wanted a rule that said you can't use a placebo if there's an existing drug. They just didn't want to hear anything else. And in fact, the Declaration of Helsinki was modified to say exactly that. There was so much controversy over this. Now, eventually that was rolled back, and the Declaration of Helsinki now says basically what most people who've really thought about this believe, that it's not ethical to use a placebo if you are, if there's an existing drug that's known to save life or prevent really bad things from happening.
But it's okay in the cases of,
but it's
drug that's known to stop a stuffed nose.
That's right. That's right. So anyway, this was a long process, and I was involved in giving talks. I'm sure I gave one at Primer about this. I probably was involved in a debate about it. I wrote papers with FDA colleagues that appeared and explained what the issues were, tried to explain the scientific difficulties in being able to draw conclusions about an effective drug when you didn't have a placebo arm. There were many people who were saying, well, if you've already got an effective drug for allergic rhinitis, say, then the only thing we want is a better drug. Why would we care about another drug that's just as good? If it's better, then you don't have any interpretive problems, and that's right. But there's ways to be better other than being more effective. You can be better by having fewer side effects. So for example, we may have had effective antihistamines, but they were sedating, make you sleepy. That's not so great. You may not want to be sneezing and blowing your nose all day, but you don't want to be half asleep all day either. So the creation of non-sedating antihistamines was a huge step forward for people who had allergic rhinitis. Now, these antihistamines were no more effective than the sedating antihistamines. In fact, maybe they were even a little less effective. Who knows? But, people who needed this was really great. But you couldn't have determined whether it was effective at all without a placebo control.
Right.
So this was something to explain. And there were people, I will tell you, who were like this. Don't tell me. I just know it's unethical. I don't want to hear any arguments. I went to a lot of meetings where there were people like that. But ultimately, people did hear. At least enough people did hear. And the declaration moved back. And I think we're pretty much back to where we were.
Do you think they came around because just reason came to light eventually, or do you think it had to do with how the trials in really dangerous, life-threatening situations were improved, so they stopped feeling so worried about it, or some other reason?
I think some people are not convinced. I think the Institute of not the Institute of Medicine, the World Medical Association had yet another discussion of the Declaration of Helsinki, and I'm sure that the people who were unhappy that it was that this particular placebo point was turned around are still arguing about it. I think there were a lot of people, thoughtful people, who initially took the very hard line that placebos were always unethical if there was an existing treatment. I think finally kind of heard the message and softened that position. I think that did happen. But I don't think everybody's persuaded.
Right, I guess not. Susan, we've just touched on, I see that you've had a long and very varied career, and we've only touched on some pieces of it, but I wonder if you look at the big picture, can you see a trend of how your professional sort of priorities and goals have evolved in the time that you've been doing this work?
Well, I've really never given that a lot of thought. I've sort of taken one step at a time.
Right.
I'm always interested in learning something new, and in each new position I've taken, I've been able to take what I learned from before and see how it applied to the new thing. But being new at it, then you can sometimes look at things in new ways. When I went to the FDA from NIH, one thing I had to deal with was product safety. I was now head of an Office of Biostatistics and Epidemiology, and at FDA, epidemiology mostly meant post-marketing safety surveillance, and I was in the Center for Biologics, which had responsibility for vaccines, and there's, having to deal with all of the concerns about the safety of childhood vaccines, where the clinical trials didn't give us much of an answer, because, if there was a serious adverse event, it would be very rare, and you would only find out about it in post-marketing surveillance. But the post-marketing surveillance data are very difficult to interpret. It's just the worst kind of data you can possibly imagine. And so trying to see what we could make of the data that we have, and try to understand what get more of the understanding about what the clinical trials were telling us, and whether the clinical trials were telling us enough. Those were interesting questions. So I'd never worked in vaccines before, but that was a whole new area for me, and the area of post-marketing safety surveillance was something that I hadn't thought about before. So I've been able to, think about things maybe in somewhat new ways. One thing I was very adamant about when I was at the FDA was reporting what we found, and that was not so standard at the FDA. People at FDA occasionally wrote journal articles, but we actually tried to look at the data that we had and write it up and report it to the public to provide some assurance that in fact somebody was looking at these data and that somebody did care about whether there were post-marketing safety problems. So that was interesting too. But I can't, it isn't that I've ever really consciously thought about how things were evolving. But like I said, each new job brings something new that you can bring your prior experience to.
And it sounds like the experience of tackling something new maybe was part of what was satisfying.
Yes. Scary in the beginning, always. We're okay.
Yes.
Yeah.
Well, we are drawing to the time when time is short. And so I'm wondering, I know there's many topics that I haven't touched on, but are there issues of particular interest to you that you'd like to bring up and discuss in our remaining time?
Let's see. We've talked about the AIDS issue. We've talked about the placebo issue.
Or we could revisit those even.
We got a little bit into the vaccine stuff. One of the concerns that I had when I was starting to look at the childhood vaccine issue was how much data do we actually get from clinical trials, and whether our clinical trials of new childhood vaccines were large enough. That's a hard sell, because vaccines have been such a wonder in protecting children from the horrible diseases of the past, that people who work in vaccines are very reluctant to think about, if we can decide that a new vaccine is effective with a study of 500 people and get that vaccine out there, how is it possibly ethical to think about saying we need a much larger study to just see whether there's some rare reaction?
And so the problem with the larger study is that there's therefore a larger subset of people who are not getting treated by it, it
just takes longer. It takes longer. If you want to do a study of 20,000 children, it's going to take longer than a study of 500 children, and that means that it's going to be that much longer for the vaccine to get out there. So those were some things that we discussed initially internally and eventually externally, and I think there was not. I didn't really get too many people on the inside to think of this was very much of an issue until the first rotavirus vaccine came out, and within a year had to be recalled because it generated cases of not commonly, still rare, but it was pretty clear that these cases of intussusception, which is the bowel telescoping in on itself and it can be life-threatening, although I don't know that we had any deaths. Usually, a child gets into extreme pain with this, and so the parents rush the child to the hospital, and the child can be treated. But it was certainly not considered acceptable that there should be a universally given childhood vaccine that had a side effect like this, and it was withdrawn. And I think that did get some people to start thinking about, because that vaccine had been approved on the basis of clinical trials of just a few thousand people. And it did get people, I think, started thinking about how much data we really did need in the randomized fashion, where we had controls to really see whether we had a problem. More recent new childhood vaccines, the trials have been much larger, not entirely, in fact, maybe not even at all, because of concerns about safety issues, but because the disease these were intended to prevent, the infections, were more rare, and so you needed bigger sample sizes to be able to see them.
And so you were on the side of advocating for
larger trials, yeah.
And again, do you have any wisdom about what it took to? Did it take some adverse events happening to bring people around to that? Or
No. I'm not. I would not say I brought people around to that. Okay. This is one of my unsuccessful soapbox things. But what I'm saying is that. People are at least open to doing larger. The trial of the second rotavirus vaccine had almost 70,000 children in it, but it was to look at the safety issue. But there they recognized that it was essential. There was no way another rotavirus vaccine was going to go on the market without being able to rule out that it had an excess number of cases of intussusception. So when it may have been before these discussions, maybe nobody would have thought that was doable. And I think raising consciousness about sometimes needing that amount of data was maybe helpful in encouraging a company to start a big trial like that. But other studies, like for example the Prevnar vaccine, the pneumococcal conjugate vaccine, meningitis vaccine, those were all studied in larger sample sizes because you needed those larger sample sizes in order to observe the outcome. For the pneumococcal conjugate vaccine, the outcome was invasive pneumococcal disease, and that's quite rare. So you needed a big sample size to look at it. But then you had the safety data because you had the larger sample size. So that was sort of an interesting experience. And I mean, it's very important to be able to persuade parents that a vaccine to be given to their children is going to do them more good than potential harm. People are worried about adverse events, but it's one of those things that it's very, very, very rare to have a serious adverse event caused by a vaccine. What's not rare is that something bad might happen to a child after they got a vaccine. If you're vaccinating every baby, starting at birth and then at 2, 4, and 6 months of age, any bad thing that's going to happen to a baby is going to happen after a vaccine. And some of those are going to happen within a few days after a vaccine, just by chance. But you can understand that if you're a parent and your child, something new, something bad happens within a few days of a vaccine, you can understand that a parent is going to be suspicious that the vaccine might have had something to do with it. But, you can say in advance that there are going to be a certain number of those cases. So it's difficult. Dealing with that was a lot more difficult than dealing with the AIDS activists.
Really?
Oh, yes. Because I think the anti-vaccine movement is largely consists of people who have had an experience, they or someone close to them have had, something bad has happened to somebody in their family, and they're convinced that the vaccine was the cause, and then this is what they devote their life to, trying to see that this doesn't happen to other children. They're very sincere and very well-meaning, and many of them really are very knowledgeable about vaccines, but the science just doesn't bear out what they're saying.
Well, Susan, I didn't mean to put words in your mouth by saying that you succeeded in this soapbox endeavor, but I'm wondering, that makes me wonder, when you look back at there's several issues on which you've taken part in some kind of a controversy. Are there parts of your career that you think of as your legacy achievements that you're particularly proud of? Not to say that there are not going to be many more, but I'm just curious so far.
I feel like what I'm proudest of is that when I would see something that I thought was a problem that somebody could do something about, that I tried to do something about it. That I think not everybody might take that road, and in many cases I feel like I was reasonably successful in what I was trying to do. But I think it's a question of, this is my job, here's something out here that happened, gee, maybe we could fix that. But if I don't do that, if I don't go after that, which is going to be difficult and controversial, nobody's going to expect me. I'm not being assigned to do it. Nobody's going to expect me to do it. But so I won't be in trouble if I don't do it. But maybe I can try and do something about that. And I think that's what I feel good about in my life. Look, finding things, seeing things that I thought maybe I could do something to help, and giving it a try anyway.
Great. Thank you so much, Susan, for taking the time. I really feel privileged to have heard your story, and I'm also really grateful on behalf of Primer that you took the time to contribute to this archive that we're beginning just now.
Well, I've enjoyed it.
Thank you so much.